A'udhu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. So today we are going to have today we're going to start the next chapter of animal biotechnology. And the first thing that we are going to discuss about is cell culture. This is the online lecture number 8. And Alhamdulillah, we are cruising through the course very good. And I really appreciate the way you students are responding to the recorded lectures, asking questions through the 30 minutes interactive session, which never is 30 minutes for us because we have so many queries and so many questions of so many students that it always goes over 30 minutes, which is really good. And now we are going into the sophisticated part of the course, starting from bacteria, jumping towards plants. Now we are going to animal biotechnology, and I am following this pattern because this would simplify things. If you have uh, know about genetic engineering, which was mostly based on bacteria and other prokaryotic microorganisms or organisms, then we jump to plant biotechnology. We also talked about genetic engineering, which results in the transgenic plants production in higher eukaryotes because plants are eukaryotes, and then we talked about tissue culture. Now we will jump to animal biotechnology, and animal biotechnology is going to be a longer chapter. It will be divided and further subdivided into different parts, but we will start with the basic cell culture. Here we will give an overview of cell culture, and we will also give some overview of microbial as well as plant cell culture, which we already discussed. Not the microbial really, but the plant culture, cell culture we had discussed in very much detail here in this part of chapter of plant biotechnology. Then the production of transgenic animals in the next lecture, not this one, but the next lecture. And then on the online lecture number 11 would be about stem cells. So one of the ways of production of transgenic animals is directly linked towards stem cells. It was a little tricky if we should teach this topic before animal production or animal production before stem cells. But then I think we can blend it together when we have discussed number 10 and number 11. In the end of lecture number 11, I will blend it together. Hopefully we will have a good idea of understanding it. So without any further delay, let's start today's lecture about about cell culture. So if we talk about mammalian cell culture, again, this lecture is mostly emphasized on bacterial and mammalian cell culture because about plant cell culture we had discussed in detail in the plant tissue culture. The ability to grow cells in culture, it means we have isolated, we have harvested, we have excised cells or tissues, and we grow them inside a lab under controlled environment. This is known as a plant, or this is known as a mammalian cell culture. Generally, any cell, either a plant cell or a bacterial cell or a mammalian cell, when we culture it in petri dishes or petri plates or flasks, even these days some scaffolds, with a controlled environment, giving them all the growth nutrients for a number of purposes. As mentioned here, biotechnology and research. So biotechnology here represents for the production of important biochemicals like proteins, like vaccines, like growth hormones. So this is known as cell culture. I have placed the applications of cell culture before everything else. So we talk about bacterial cells. Gene manipulation is one of the major applications of cell culture. We use a number of vectors and the plant Why do I keep on saying plants? I don't know. Sorry for that. So we need the gene manipulation, which means the number of plasmids has to be multiplied. Any kind of plasmid, we have to multiply it. And bacteria is the biological factory for that. Similarly, we need to keep cDNA as well as genomic DNA libraries. And again, for genomic DNA and cDNA libraries, we need to go to bacteria and hire their services. And if we want to culture bacteria, we want to have cell culture. This is one of the biggest advantages of cell culture application. Now we designed the next application is if you design some drugs. For example, these days, COVID-19, SARS, coronavirus 2 pandemic is all around the world. That's why we are having this online lecture rather than face-to-face lectures in the lecture hall at Comsas University, Islamabad. Many groups are working about designing new vaccines as well as drugs. So if we have a drug designed for, let's suppose, any kind of disease, you want to check the effects of them on the mammalian cells. The toxicity issues is an important issue because when you apply the drug, you are not only concerned that the effective or the causative agent, whatever it be, is dead, it kills it, but it also should not affect the normal cells. And this is another aspect of applications of cell culture. Producing new plants, as we discussed in detail in the plant biotechnology lecture. Agriculture for the production of silage, for pharmaceuticals, because genetically engineered bacteria, one of the classical example is genetically engineered bacteria can produce insulin. It was very problematic before genetic engineering, before cell culture. Because if you want to produce insulin, you want to use genetically engineered bacteria, but you want to culture them, and then you purify insulin from there. Insulin is an old example. A number of growth hormones, a number of vaccines, they are produced in cell cultures. Food production, baking and brewing, this is very old, and for this you always need cell culture. Sewage treatment, when we have our wastewaters, you want to treat it. And for biodegradation, again, bacteria and yeast and other fungal cells are important players in this process. So if you want to have sewage treatment through the biodegradation process using these microorganisms, we need to culture them, cell culture. So that's the application of cell culture. So just some major applications of cell culture. What are the important conditions needed for cell culture? As we discussed already in detail in plant tissue culture, the growth medium is very important. Growth medium contains the carbon source. Growth medium contains the macronutrients, the micronutrients, the growth factors, the growth enhancers. All these things are in the growth medium. And the type of the growth container and the fermenter. If you are going for a fermentation process or you're going for a normal batch culture, yeah, also it's very important. Temperature, because you have to also always go to the optimum temperature, the optimum pH, the gaseous exchange. Now, what is meant by gaseous exchange? I can tell you about some cells of bacteria. They would love to grow in high amounts, high concentrations of oxygen. Some would never tolerate oxygen at all. They are totally anaerobic. Some would love to have a bit of oxygen, the microaerotolerant or the microaerophilic. So the gaseous exchange is very important. The aseptic conditions, very important. If you cannot work under aseptic conditions, contaminants can find their way, because the growth medium is a lot of food supply. So you want to have the conditions aseptic, sterile, so that only the candidates cells survive and grow without any interference from the contaminant cells. And some methods of monitoring the cell growth. depends on the type of culture, the type of goals. You have these cell growth monitoring strategies and safety measures and applications, because you do not want to spread your cells all around. You cannot flush them out. These days, in our country, Pakistan, everyone is saying, why aren't the universities doing this, helping Pakistani hospitals, the clinicians, for the diagnosis of coronavirus 2019, the SARS coronavirus 2 detection, because it's just a simple reverse transcriptase PCR mechanism. But the problem is here, because to deal with coronavirus 19, you need to have a BSL level 3 laboratory. And I think there are very few BSL 3 level laboratories in Pakistani universities. So that concerns the safety measures. Like, we can do all the things even in ComSats in our labs, but we do not have the safety measures and applications. What if the contamination goes out? So this means if you're working with some kind of tricky or dangerous cells, maybe virus, maybe cancer cells, maybe some neurons, maybe bacterial cells, candida cells, and if you do not have the safety measurements, it's gonna be bad. It might look really, really bad. So we have to have safety measures and we have to imply them. So a really strict implication is really important to contain the stuff inside the designated labs. We already discussed this, so we want to avoid contamination of growth medium cultures. All inanimate. Inanimate means non-living. While you can also include the crystals of viruses as inanimate. So, and living objects, this could be the spores of bacteria or the bacterial cells or fungal spores. Yeah, a number of airborne spores are flying around, mostly fungal spores. So they can really contaminate your cultures. And there are techniques how to maintain aseptic conditions. Sterilizations of all the utensils or the labware using heat. You know, we use autoclave, where we go to 121 degrees centigrade with a high pressure that kills almost everything. Archon of bacterial spores. The growth of pure cultures is very important. So if we just maintain pure cultures, we would have less chances of contaminations. Okay, microorganisms, they are everywhere and they are highly adaptable to surrounding environment. About bacteria and about microorganisms, there's one very common or very important phase. They are designed to adapt. Nature, Allah subhanahu wa ta'ala, has produced them and gave them the capability, no matter what conditions come across you, you will find a way and survive. And this is true. For almost every antibiotic, they have found a way. Sometimes it takes them time, but eventually they come up with a solution. Why? Because naturally they are programmed to survive. Their only goal is to survive and divide. A bacterium does not want to do anything else but to become bacteria. What did I say? A bacterium just wants to become bacteria. It means it wants to survive and divide, or simply just want to divide. But if it cannot survive, how will it divide? So they are naturally programmed and they are everywhere. They're very easy to culture. Very easy to culture. I colonize in really unimaginable conditions. You can find them in glaciers, and you can find them in high temperature hydrothermal vents in the ocean bed floor, where there is a lot of soil, salt, high salinity, and you can still find them. They are surviving there. in the boilers, in the geysers, or geyser, I don't know how to pronounce it. You see, they survive everywhere. There are two types of microorganisms. One is the unicellular algae or the phototrophs. We call them blue-green algae, but blue-green algae is actually not an algae, it's a bacterium. And they have a vital role for the provision of oxygen in the sea. You know, the biggest forests in the world are Amazon. Amazon is the largest forest in the world, but many times more oxygen is produced by these unicellular or blue-green algae in oceans than Amazon. So they also have a very major role in maintaining the ecosystems of the sea, oceans. And then there are some heterotrophs. They need media complex containing organic source. They always need an organic source and other compounds like amino acids. Not always, but sometimes they need amino acids with micro and micronutrients. As we discussed, for food industry, you want cheese, you want baking products, for example, a bread, for example, what else? Baking products. All these cheese, the dough rising breads. Wine and beer, we do not really use them, but these two are really huge industries. Chemical production, for example, acetone, for example, ethanol. You know, for example, ethanol for fuel purposes, I could say, again, because we are avoiding alcoholic drinks, it's not allowed in our religion, it's haram. But if you talk about ethanol as a fuel, yeah, why not? The basis of food chains, they make the basis of food chains. This is where the food chain starts from. So they play a very important role in shaping our ecosystem. Commensal bacteria in your digestive tract. Yes, there are a lot of bacteria which releases important chemicals to help your digestive system to let the food digest. And therapeutic compounds, insulin, some vaccines, or hormones, for example, for hemophilia patients, as well as for forgot the name. For the hepatitis C, interferons, very important part of our therapeutic compounds. They're all produced by bacteria and through cell culture of bacteria.